going to study three important experiments which led to the step-by-step discovery of DNA as a genetic material. So at 1900s there was question that what is hereditary material. Scientists initially believed that proteins could carry hereditary information because they are highly complex, built from 20 different amino acids that allow immense diversity. In comparison, DNA seemed too simple with only 4 bases. It appeared insufficient to encode the complexity of life. This is why proteins were first considered the stronger candidate while DNA was underestimated. So now we comes to the first experiment, that was Frederick Griffith's experiment. He was not intentionally doing this experiment for the discovery of transformation, but he was working actually on a very serious practical public health problem. And what he was doing, he was a bacteriologist in the UK in 1920s. He was mainly studying the bacterium Streptococcus pneumonia, which causes pneumonia. So at Griffith's time, there was a disease that was pneumonia that was the major cause of the death at that time. That disease was being investigated for the treatment, and scientists were trying to develop vaccines and understand different strains of bacteria. So Griffith was investigating that why some strains of bacteria are virulent and others are harmless. How the body develops immunity against these strains. He worked specifically with two forms. The first were S strains. S means smooth, that bacterial cells were having capsules and they were deadly, mean pathogenic. And the second form was R strains, which was rough strain. They were not having any smooth layer, that was capsule, and they were non-deadly, mean non-pathogenic. So what was the question that led to the transformation principle? The Griffith's curiosity was something like that: can we use weakened or dead bacteria to create immunity against dangerous strains? And this idea was actually very similar to early vaccination concepts where killed or weakened pathogens are used to safely train the immune system. But what he discovered went far beyond immunity. It hinted that something from the dead bacteria could actually change living ones. This is the diagram of the Griffith experiment, and in this diagram you can see different types of experiments he did. And in the first experiment he took R strain that was non-virulent and injected that into the mouse, and mouse remained alive. In the second experiment he took smooth strain that was virulent, and when it was injected into the mouse died. In the third experiment he used heat-killed S strain, and it didn't cause any disease and the mouse remained alive. In the fourth experiment, he used rough strain alive and heat-killed smooth strain dead, and the result was very unexpected, that mouse died. He thought that when I used heat-killed smooth strain, the mouse remained alive. When I used R strain alive, the mouse remained alive. But when I used a mixture of both, why the mouse died? This was the point that led him to think that something was there that was transferring the information from dead S cells to live R cells, and he also collected live S cells from this dead mouse body. Concluded that some transforming principle from dead S bacteria was taken up by live R bacteria and converted them into deadly S type. He didn't know it was DNA yet, but this discovery laid to the foundation for an important discovery, that was the discovery of DNA, that is the genetic material. Now we comes to the second important experiment which was done after the Griffiths experiment, and this was Avery, MacLeod and McCarty experiment in 1944. Griffith's experiment clearly showed that some transforming principle from the dead S bacteria could turn harmless R bacteria into deadly ones, but he didn't know what the substance actually was. This unanswered question caught the attention of scientists like Oswald Avery, Colin MacLeod, and MacLeod McCarty. They wanted to go a step further and identify the exact molecule responsible for this transformation. So, building on Griffith's work, they designed an experiment to test whether this transforming principle was protein, RNA, or DNA, bringing science closer to discovering the true career of genetic information. Question still was the same, either hereditary information carrier is DNA or protein. This is the experimental setup, and this was the hypothesis they devised, that the genetic material of the cell is either protein, either nucleic acid, that may be DNA or RNA. So what they did, they took a test tube and took a sample of the dead S cells. Dead S cells. S mean smooth, that are virulent, that are pathogenic. Dead S cells. In these dead S cells, because these cells are dead, they are having lipids, sugars, proteins, DNA, RNA. So they removed lipids and sugar because we want only test that either protein is the genetic information carrier or DNA or RNA. So this is the first step, in which they remove lipids and sugars from the solution of heat-killed cells, and proteins, RNA, and DNA remain in the solution. In the second step, they treat the solution with the enzymes to destroy protein, RNA, or DNA. And in the third step, they added that they added these test tubes into a culture containing living R cells and observed for transformation by testing for the presence of virulent S cells. Now we understand the experiment in this diagram, that they took first of all this test tube, and this test tube was having dead S cells, their lipids, their sugars, their proteins, their RNA, their DNA. These two things were removed. What was left was DNA, RNA, protein. So they took three test tubes. They took a small portion of the solution into this first test tube and added proteinase enzyme to this test tube. So what happened? Protein in this test tube was destroyed. No protein in this test tube. Now they put this culture or this solution into a flask which was having R cells which were alive, and they observed that this flask, after some hours, there appeared live S cells. What was there in the flask? There were live R cells, and there was DNA and RNA of the dead S cells, which was added from this test tube into this flask. So they thought that either DNA or RNA is carrying the information which transformed these live R cells into live S cells. So now we comes to the second test tube, in which they took a small culture from this test tube into this one and added RNase enzyme. RNase destroyed all the RNA in the test tube. What left was protein and DNA. So when they added this solution into the flask which was having already R cells alive, they noticed that in this flask also S cells appear after some time. So it means something was there still that was carrying the genetic information after destroying the RNA. Now we comes to the third test tube that was having protein, RNA, and DNA. We added DNAs. No DNA was there. DNA was destroyed. So this solution was transferred to the flask that was already having live R cells. So after transferring this solution to this flask, scientists observed that no transformation occurred. No S cells appeared here. Only live R cells were there. So they confirmed that the DNA was carrying the genetic information, because when it was destroyed, no transformation occurred. Otherwise, during the presence of proteins, RNAs, there was no effect on the transformation. So they concluded that when proteases were added, transformation occurred. When RNAs were added, transformation occurred. But when DNAs were added, no transformation occurred. So they concluded that DNA is the genetic material because only destruction of DNA stopped the process of transformation. So the conclusion was DNA is the genetic material. Even after the landmark finding of the scientists like Oswald, Avery, Colin MacLeod, and MacLeod McCarty Experiment, many scientists continue to debate their conclusion that DNA was the genetic material. At the time, proteins were widely believed to be the carriers of genetic information because of their greater structural complexity. As a result, the idea that DNA, a relatively simple molecule, could store genetic instructions was met with skepticism. It was only after further experiments, such as the Hershey Chase Experiment, that the scientific community widely accepted DNA as the hereditary material. Now we are going to study the most important experiment, that is Alfred Hershey and Martha Chase experiment. This experiment provided the proof that DNA is really the genetic material and it is really the hereditary information carrier. And this experiment is also known as transduction principle. Transduction principle. As we have studied the Griffith experiment, that was also known as transformation principle because the definition of transformation is that uptake of genetic material by bacterial cell through the environment. Because the S cells were dead and their genetic material was transferred or uptaken by the bacterium that was R type, that was kind of transformation. And in this experiment, the genetic material is transferred to the bacterial cells but through a virus. So virus mediated genetic material transfer is called as transduction. That's why this experiment is known as also transduction principle. So what they were doing before the experiment? These scientists were virologists and they were studying bacteriophages. Bacterio mean bacteria and phages mean eater. These are those viruses which eat bacteria, mean they infect bacteria and rupture the bacterial cells, releasing new bacteriophages. They were studying these viruses. These are viruses. And they knew that these viruses are only made up of DNA plus protein coat, and you know the structure of bacteriophage is like this one. These are the tails and this is the protein coat, and inside the protein coat there is the genetic material of the virus. So they thought that bacteriophage are only having DNA and protein coat. DNA and protein coat. So, because the previous discussion among the scientists was that either DNA is carrying the hereditary information or protein or RNA, so they thought that if in some way we can label DNA or protein or both and can track the way they enter the cell during the infection, so we can actually determine whether DNA enter the bacterial cell or protein enter the bacterial cell, because whatever will enter the cell, that will be having the carrier of genetic information. So they were having this curiosity that when a virus infects bacteria, which part actually enters the cell, and that part should be the carrier of genetic information. Hershey and Martha Chase thought that if we can track the way of DNA and protein during the infection of bacteriophage to the bacterial cell, we can see which one enters the cell. So in this way, we will be having a confirmation that either DNA enter the cell or protein enter the cell. So when whatever will enter the cell, that will be the information carrier. So they used radioactive labeling. They labeled DNA with phosphorus. They labeled protein with the sulfur. And why this labeling worked? Because DNA is having phosphate group and proteins are having disulfide bridges. And in this way, DNA contains sulfur but no DNA contains phosphorus but no sulfur, and proteins contain sulfur but little or no phosphorus. So in this way this labeling worked, and this labeling was radioactive. Sulfur phosphorus 32 was also having radioactivity. Sulfur 35 was also having radioactivity. So it was very much easy to track the way that either proteins are entering the cells or either DNA is entering the cell by recording the spectrum of radioactivity of these radioactive atoms. This experiment contained three steps. First was the infection, second was blending, and third was centrifugation. In this diagram, you can see two groups of bacteriophages. One is having sulfur labeled protein capsule and the second is having phosphorus labeled DNA core. When scientists allowed these bacteriophages to infect bacterial surface, these these viruses infected bacteria, they injected their DNA into the bacterial cell and their protein cores were outside of the bacterial cell. So scientists wanted to detach these viral protein coat from the bacterial cell surfaces and they used the step of blending through a machine that is blender. This blender agitated these cells and removed viral protein coat from the bacterial cell surfaces. In this way, both the test tubes were having the mixture of viral protein coats and bacterial cells. In the third step, that is centrifugation, scientists wanted to separate viral proteins and the bacterial cells. So these steps separated the viral proteins and the bacterial cells, and when centrifugation is done in a test tube, you can see on the screen that this is the diagram of the test tube, and whatever remains in the bottom, that is called as pellet, and the upper portion is called as supernatant. So bacterial cells were heavier, they remained in the pellet, and the viral protein cores were having a lighter weight, so they remained in the supernatant. And scientists recorded the radioactivity because this portion was having bacterial cells were having DNA, and through this portion, DNA was labeled with phosphorus. So radioactivity of the phosphorus was recorded from the pellet, and radioactivity of the sulfur was recorded from the supernatant. So in this way, this experiment proved that whenever bacteriophage will infect bacteria, it will inject its DNA to make more bacteriophages. It means genetic information is being carried through DNA and not protein, not RNA. As we have seen from the beginning of this lecture that our first experiment of Griffith provided a clue that something is transforming principle, that may be DNA, that may be RNA, that may be protein. Our second experiment of Avery, MacLeod and McCarty, it provided experimental proof that DNA is really the genetic information carrier. And third experiment of Hershey and Martha Chase, it confirmed that DNA is really the genetic information carrier. So in this way, DNA was proved that DNA is really the genetic information carrier. So now attention of the scientists moved from is the DNA genetic material to how does DNA work. Thanks a lot for watching.